Thanks a lot, Shane. So just to reiterate, so Renate and I at the University of Pretoria, University of Pretoria Genomics Lab, and we also wear other hats. So both of us are part of FABI, and in this talk we also want to acknowledge 1KSA and the DTI for their assistance. All right, so a little bit of background. So Renate and I have been sequencing since 2012. We've done a lot of ONT iron torrent sequencing, but we made contact with Diplomix in, I think, 2022, and we officially started the UPGL in 2023. All right. So it started off with Renate and myself. And luckily from this year, we've also been able to have Candice Deval join us as a technician. We were officially started in 2023 during our first NGS Open Day. It's now become a tradition. It's been attended by many researchers and students from all over Africa, Middle East, and also in all the academic institutions in South Africa. Our next one is in end of June at the 27th, and we expect a similar upcoming. So I'll talk a little bit about workshops at a later time. As part of our facility, we have a P2 Solo, and we also have access to another P2 Solo that we use from the FMG group at the University of Pretoria. And as part of our method development, we started with two different species. So the one was the pangolin. The idea was to sequence 6 individuals from 3 ecotypes in South Africa, and this is to assist, first off, to assist the rehabilitation of rescued animals, and then also to assist the police, because once, let's say you've captured a lot of scales, if you can then have the genome sequence so you can get markers, so that we can identify smugglers and arrest them and go through the whole legal processing. The second method development project was the South African, the real yellowwood, and this was also to help us to get some DNA isolation expertise, because with the yellowwoods there's a lot of resin involved there. So these two formed the basis of us getting started with the facility and getting our hands wet so that we can offer a service also to other researchers. Part of this, again, was DNA isolation methodology. So we had several different kits we wanted to try. So for the pangolin project, we identified the Magrinagel nucleobond kit, the nanobind kit from PacBio, the genomics tip from QIAGEN, and then also the Monarch tissue kit. Of these, we found that the nanobind kit gave us very, very good results, and as well as the MN nucleobond kit. These we used for the pangolin samples. The pangolin samples have other problems associated with it, which is probably going to be more prevalent once we start sequencing more samples from biobanks. Because these samples were all stored in ethanol, there's some DNA damage and alteration that happens, and we've just found that we can't sequence these samples. So we are currently in the process of getting fresh samples. A lot of permitting and politics are involved there, so it is really difficult. We've put the project on the back burner, although we're trying to get past this all the time, and maybe we found a few ways of doing this. For the plants, we've luckily have access to very high quality plant research at the university, and we've partnered again with the Forestry Molecular Genetics Department. So they've developed some DNA isolation protocols that we've been able to make use of. They're going to put that on Zenodo very soon, and then we will make it available to everyone. And for this initiative, we used the Magrinagel kit again. We used the nanobind pan DNA kit, and then also the Sativa kit. We tested these out, and we have now settled on the nuclear isolation protocols from the FMG, which we then take into the Machrinagel nucleotide kit. So we've used these for several different species. The sequencing for the yellowwood is complete. The DNA was very clean, sequenced very well. And then we also used that for the Greya sutherlandi species for the 1KSA. Traditionally very difficult, also has a lot of polysaccharides, but we could get a really clean DNA and the sequencing went very well. So for us, we have together some methods and protocols that we can suggest to not just service clients, but also to 1KSA submitters, and that we can also assist them with in the lab. In terms of bird blood, we have several bird species outstanding. We're working with the researchers there. And together with Sandy, we've now come to suggesting the PureLink genomic DNA kit that worked very well with their bird species. We're trying it out with some species of ours to see if we get good enough DNA out that we can actually sequence and go ahead, especially with the bird 1KSA initiative that we're looking at. As you've seen with other workflows up to now, shearing is a problem. It's something that's important to do. And we have looked at two different methods of shearing here. We looked at the enzymatic method using the shear-raise plus from Zymo, and then also the bead shearing method that Nanopore has on their own website. So we've tried both of these with different measures of success. The bead shearing, for some reason on the yellowwood, did not work at all. The enzymatic method did work. So we have now put together some timings. It's based on the amount of DNA that you use, the concentration, and the number of enzyme units you use. We use the shear-raise to bring the fragment sizes down to a manageable level where we can then go further. Also with this, we've incorporated a short fragment removal, where we can either use the PacBio kit if we want to remove fragments that's mostly smaller than 10 kb, and then the Nanopore short fragment removal kit where you can remove things mostly shorter than 20 kb. So it all depends on what your input DNA is like, what those sizes are of the small fragments you want to remove, but it depends on your DNA concentration, also the amount of DNA. So similar to the decision tree that Anya has put together, we kind of have a bigger decision tree, where we look at exactly what we have coming in and how we can then get the best output out. Right. Which brings us to shearing and N50s. So we've been struggling a lot to get longer N50 values for our sequencing. It's if we look at the Crozet shag, for example, this is one of the genomes that we've just completed. Even though we have a library that was built that has a 14 kb size, when we sequence this, we still only sit with a N50 of about 5. We really want to improve on that. What we found here is that, unfortunately, the technology that just about all of us use, TapeStation, is seriously unreliable, because we're also sitting in that range that even they say is the gray area for TapeStation. I would think up to about 12 kb, it's very accurate. Over that, it's a coin toss. So by using comparing TapeStation with fragment analyzers, we've seen that the TapeStation overestimates your DNA fragment size between 2 and 3 times. Which means that previously we've always tried to target fragments or library sizes in a range of about 20 to 30. We can't do that. If we do that and we overestimate it twice, then we are sitting with fragments for 14, we're sitting at 7. 7 is in the range of 5. So I think we use that as a rule of thumb, at least 2 times it overestimates. And we tested this out on our yellowwood samples. One run, I can't exactly remember the size of the library, but our fragment size was about 5 kb. We get very good yield out. We had 150 gigabases of data, but only had about a kb of N50 of 5 kb. For this run, we used DNA that from sharing results, we were sure that the fragment sizes were just over 60. The cutoff for the tape station is 60. So we sequenced this, and we could get 90 gigs of data, 90 gigabases of data at an N50 of 35. And this is ideally where we want to be. So we are very excited about this specific run, because now we will put it through the Hi-Fi ASM protocol or assembly. The Yellowwood genome sits between 6 and 8 gigabases. We want to see if this specific run is going to be enough so that we don't have to give more data at this range. And we're hoping that it is. But we still have about 150 gigs of data at an N50 of about 5 KB that we can use to supplement that assembly, and then also use as a polishing data set for this assembly. So we're really interested in specifically this one to see how it goes forward. This means that we are now altering our decision tree. We take the tape station as a kind of a guidance, but we don't trust the values. We work at least 2 or 3 times difference in the value you get and the value that you really have. So for our decision tree now, we would go on working on how much DNA you have, what the tape station says that fragment sizes are, then incorporate some kind of short fragment removal to get rid of the really short fragments, and then we can hopefully get the N50 values up into the range that we really want. We found that nanopore is not a long-read sequencer, as Tim has also said a long time. Nanopore loves short reads. So if we give it short reads, we will get 200, 250 gigs of data. So our goal is to get reads in a similar line to this, where we have a lot of DNA with long reads, and then that hopefully will give us a very good assembly. We tested this on the Greyer sutherlandi species that we sequenced recently. The DNA fragments, I think, sat at about, if you can help me, 30 to 40 KB. Our N50 was about 13 KB for our final read. So we now are going to be much less strict with our sharing. We will try to find or shear just to below 60 KB on the tape station, and then we can be sure that we will sit more in the range that we are looking for. So it was a lot of heartache and pain getting here, but I think we have a handle on this. We've done method developments on the nanopore libraries as well. This is going into Zenodo soon. First off, input DNA. We don't use micrograms at all. We go with the tape station, and then we rather work out how many femtomoles of DNA we have, and use that as an input. So we use 500 to 600 femtomoles input DNA. Once we've started doing that, we find more consistent results, and our libraries are of much higher quality. We've coupled that also to the reagents we use, because the third-party reagents are quite pricey. So we have 3 levels of input DNA or input reagent selection. Either we use the normal LSK procedure that does use quite a lot of the polishing and the end prep enzymes. We also have a half an LSK where we can use half of those reagents and we adjust the volumes, or we use the NBD, the barcoding kit amounts. So depending on what your DNA concentration and amount is, we can actually use, we found that you can interchange that first step because it's independent from the nanopore reagents. So if you have a high concentration, high amount of DNA for LSK samples, you can actually use the NBD protocol. You're going to use a lot less of your initial enzymes, and then after your first bead cleanup, you can just dilute the sample and go ahead. So we played around with this, and it actually works quite well. Then we've altered the timings for these protocols. We've extended them, especially where we have a lot of DNA. And we found that increasing the times of incubation by 2 or 3 times, all we waste is our own time, but we found that consistently this gives us very good results. Lastly, we don't use the Agent Core cleanup beads that come with the kits. We use the Agent Core beads by themselves that we purchase, because if you purchase that, the first thing they say is don't freeze the beads. So when you get it from Nanopore, it's frozen and you keep it frozen. So from when we started using these kits on our own and incorporating all of these different changes, at some points we were losing 95% of our DNA during the library prep procedure. Now we're sitting at between 40 and 50%. So we really get very good yields from our library preps. That helps us with sequencing. It helps keep the costs down. All right, so we sit at the University of Pretoria. It's an educational institution. So all of our projects are involved with student research. So the impact cannot just be measured here. The impact is measured in the student throughput that we have. It's not just educating students, it's also educating the PIs. A lot of the work Renate and I do is project planning. So we help students and researchers set up projects that's suitable for the technology, that's suitable for the question that they're asking, so that you can get data out that you can link with your project. So that's where you see more impact, not just on what's been sequenced, but on the thinking that's been changed and that capacity that's been built. These species have now been sequenced. Some of them have been uploaded. They are being base called and will be uploaded next week. So these are ready for assembly and ready for species cards very soon. You already see the crozet shag stickers that you have outside. The blackback jackal was the last one we did, and we have about 100 gigs of data for that one. So we are very happy that our methods that we've developed are bearing fruit. Challenges. Sampling, permits. Permits in red, it's always our biggest problem. I will show you the backlog now, and a lot of that is stuck with permits. For example, the pangolin samples that we want to sequence sit at the Kruger National Park. The Section 20 has expired, so we cannot move those samples. And this is true for so many of our, especially if you get to mammalian samples, sampling and moving permits, those are really big issues. So that's contributing to a lot of backlog that we have. Sampling itself, do you have the appropriate sample? For birds, just using blood spots might not be enough. That means that you will have to go into the field and collect samples. Some of them have very little DNA to give, little sample to give. So sampling and appropriate sampling is really important, especially for plants. We found that if you use very young tissue, you can immediately flash freeze them and store them at -80 until you can isolate the DNA. Makes a big difference. So together, these two have been very problematic. Then lastly, your DNA QC. Do you get DNA out of a sufficient quality and quantity that you can actually build a library and sequence, get a lot of sequencing data from it? One wouldn't think so, but fungi is not just fungi, it's not just fungi. We've had 48 fungal species on as part of the 1KSA pilot project, and we're still waiting for most of those. And it's just based on fungi growing slowly and not isolating DNA. There's a lot of, just like plants, fungi have a lot of issues with DNA isolation. So we have a set of very strict rules around DNA QC. We spend a lot of time helping researchers optimize that. Carl is sitting here. I think he doesn't like us anymore because we keep shooting down his DNA. But we've been able to put together some protocols and guidelines that allows us to, once it comes into our workflow, we are sure that we're going to get good libraries that we can sequence. One thing we've tested recently on a service project was adaptive sampling. We were very happy to do this in such a way that we are sponsoring part of the project. So just in very broad lines, we have some bovine and dog samples that they wanted to look for a certain pathogen in there. So they used some methods to subtract a lot of the host DNA, but we still sat with about 20,000 times more host DNA than the pathogen DNA. So we did adaptive sampling on the PromethION. We barcoded the two samples. The amount of DNA we got from them was especially for the canine sample, was substandard. So in the end, we loaded the bovine sample twice. So we sequenced the bovine sample, reloaded the next day, washed and loaded the canine sample, and then ran the bovine sample for a third time. And we used the genome depletion in this process, which taught us a lot, again, about Linux permissions. So it's always something to look out for. What did we find? So to do the analysis afterwards, you have to use Minimap. You take your reads, you map it back to your host genome. Use SAM tools to extract the reads that did not map. And then you can use the appropriate tools. You can use BLAST, and then you can see, do you actually find those the pathogens in your sample? This is a short video. This is super fantastic to see what we normally find when we sequence. These, the magenta spots, that's active feedback from your machine. So normally you don't want to see them because it means there's a read inside my pore that's blocking. And that's an active process by the pore trying to push it out. But for adaptive sampling, we want to see that because that's the process. If it finds a host read, it's going to kick it out. And we see here, you can see how much active feedback there is the whole time. So that is the adaptive sampling process happening in front of your eyes. So for the two bovine samples, we had, it was about, almost 45, 27, so it was almost 80 gigs of data. I got a big fright because the N50 for this is 607 bases. And I thought, oh, it's failing. Until I thought through it, and it makes sense because adaptive sampling, the first second of that sequencing is the minnow taking in the data. It uses that 400 base pairs to compare to your library, and then it takes half a second to decide if I want to push that readout. So what you're seeing here is 1.5 seconds of sequencing take up most of the data, but that's doing its job because it's taking in the host and kicking it out again. So that low N50 makes sense in the process. What we found in the end, we had 4.3 million reads for the host still there, and most of this was in the form of that 600 base pairs. Non-host was 223,000, and 81 reads comprised of that specific pathogen, which was a result because otherwise you would have to do a PCR for specific primers, to try and get these out. Now I can go and reanalyze this for other pathogens as well because they will be in there. So this was a really nice result. The same for the canine. We found a similar N50. And here we found fewer reads because just the DNA was less, but we had a lot of this pathogen in there. So in principle, this approach works. But again, we can go and reanalyze this for other samples or for other pathogens. So it's a data set that they can dip in more than once. So what's next? So I like to think of this as kind of us strolling through the kingdoms with diplomics. All of these are species that are in the queue. They've been approved. We're waiting for DNA. So Carl is sitting here. He's busy isolating red Hydropius DNA, also with his hairs. We're waiting for more St. Joseph shark. Again, it's a permitting issue and it's a funding issue to actually go and catch these. You see several very iconic bird species here. Again, waiting for DNA. Ticks. I know Anya is really interested in these. So we have a lot of tick samples. We have the flightless dung beetle that we've tried some DNA isolation on. It didn't work. We're going to optimize that very soon. And then plants. Pretoria is a very plant-heavy, our faculty is a very plant-heavy sequencing lab, so all of these species are still waiting. You can see there's a lot, also the yellowwood, the Henkeli, we have fantastic DNA out, so we're going to probably be sequencing that one next. Lucky for us, Pretoria University has one of the largest arboretums in the world. So we don't have that much issues with sampling because I can walk out my front door, Renate and I go, and we go and sequence or sample the yellowwood samples because they just on the other side of campus. So it's a very big opportunity for us to be able to sample there because we also know the provenance of it because a third of the plants on campus is part of a botanical garden. So we know the provenance, we know where they were collected. Some of it were grown from seed by our botanical garden curator. Also busy with the red grass. So this is sitting, samples are sitting on our experimental farm. So we really have a lot of resources that we're trying to get the researchers to exploit because it gives very good impetus to 1KSA and it also expands the type of questions that they can ask. We're sitting with a very strong fungal research group in FABI. These are all the fungi that are waiting, and they are more in the queue. So every time we see some researchers, they run away from us because we keep bugging them for samples to such an extent that the researchers themselves go into the lab to isolate the DNA. It's just that difficult. We have a lot of hint on the next for the next meeting of the committee. These are all the species that we are waiting for, and there will be one more there that one of our professors wants to put in. So there's a lot of flow through, but we are trying to work through the backlog in terms of sampling, permitting, and DNA quality. We do have a lot of work there. And then finally, a very big part of what we do is workshops and networking. So Renate and I had a very over-eager 2023 and 24, where we had like 7 or 8 workshops a year. Also looked at qPCR. But what we've started doing last year is we've started incorporating 1KSA in these workshops, not just in terms of allowing Shane a little bit of time to speak about 1KSA, but the samples themselves are 1KSA samples. We've had collaborators especially from Sanby, where they bring their own samples, and when they are busy with the library prep and the sequencing, they work on their own samples. This gives us a clue to what extent will this DNA sequence, because as you know, it's not a given that something will sequence. It gives us a kind of a freebie to see, will it sequence well enough? But more important, it allows us, the researchers themselves, to see what's involved in this process. I work with my DNA. What effect will it have if my library prep's not ideal, or if the DNA is not ideal? What will the library look like? When I sequence it, how well will it sequence and how well not? So it helps them also to get an idea of what they can expect. As Renate says, it's managing expectations is a very important part. So this is part of our the Africa BP workshop. It's part of the lab workshops that we do. We have our open day, as I said again this year on the 27th of June. We have a lot of fun because Anya comes with as well, and we can play in the lab. But yeah, this is kind of the core of the whole process, is because we want to expand capacity. We've been working with some of the HDIs in the region. We involve them a lot. We've been mentoring them, and we have an open invitation to them and just about anybody. They can come to our lab. We have no secrets. You can come to us. We will teach you everything we know, help you to get the most out of the sampling that you will do at your own institutions. If you have a Mark 1C, or if you have a Mark 1B or D, then you can do that. But we will help you through that process, because we also believe in that open science. If we've made the mistakes, you get that benefit, because then you don't need to spend the money and the time to learn that as well. So That's also why I just sat here and looked at Anya's decision tree, and I think we have to make that for us too, because ours is a little bit more expansive, and they have that on Zenodo as well. All right. So I just want to thank everybody on this list, especially Diplomics, for all their assistance and their help. It's been fantastic taking this journey with you. And then especially the ACGT, because they help us manage our workshops. They do a lot of work on there. We try to partner with them so that we get both Diplomics and the ACGT get benefits for all the capacity building that we do. All right, thank you very much.